unconnected like Greek mythology and this and that. But she wanted to do it here because she said she got a chance to do genetics. While she was doing her PhD, she was awarded the Rajiv Gandhi Fellowship to work under Dr. Sharat Chandra at Indian Institute of Science. Later on, when she got into the University of Michigan at Ann Arbor, both my husband and I were very happy for her because that's one of the premier institutions for molecular biology. And after that, she pursued her postdoctoral research in this retroviruses, they call it. Like I would put it like HIV virus, one of the retroviruses, and she was into avian retroviruses at that time. And that's what it just goes on. But here I have to say that her husband has been a great support, and he supported her all through to do her PhD as well as her postdoctoral research. And here I won't take much time. I think this sort of sums it all up, and she didn't want to become a doctor. And I don't regret, neither my husband nor I ever regretted that we didn't force her into do medicine, because she excelled in whatever other branch of science that she took. Thank you very much, and on to Mallika. Mr. Raj.
Mallika, it's all yours.
Thank you. Thank you for that. Thank you for inviting me to share what little I know about the virus, and we're all gaining more knowledge every single day as this virus changes. Can everybody see my screen?
Yes.
Okay. So I will have the chat box open as well. And so if you have questions, please let me know. It pulls up and I can go ahead and look at it there. So this talk is about the virus that's ravaging us, as Mr. Raja told everyone. And so COVID-19, I will briefly touch upon the virus, how the virus affects us, and also the vaccines that have either been deployed or are in development. There's a whole plethora of them. And the vaccine industry has never worked as fast or as efficiently as it has in the last few months. So to start out with, COVID is caused by a virus. So I thought I'd start out with talking about what are viruses. So viruses are pretty much non-cellular particles that contain genetic information that is wrapped in a protein coat. Now, there are different kinds of viruses depending on that genetic information that they carry. One are the DNA viruses. They have DNA as their genetic information, a molecule. And then there are RNA viruses. They have RNA as their genetic material. This includes coronavirus. And then we have the retroviruses, which are a completely different, they're RNA-based, yes, but they have a completely different mechanism of working. And so what are the differences? A DNA virus, basically DNA, when it makes copies of itself, has good proofreading mechanisms. So it's not going to change so much. It's not going to vary. And therefore, producing a vaccine against it is actually a little bit easier because they're not changing. We already have the key codes, as it were, to lock onto the virus and therefore prevent it from creating infections. And so that's how we have the smallpox virus vaccine came in, and it has smallpox has been eradicated. Now, RNA viruses are a different kind of category because here RNA, when it wants to replicate, doesn't have a proofreading mechanism. It's more of let's make as many copies as we can without reading through. So mistakes are made, and when mistakes are made, mutations happen, things change, and therefore trying to pin down a actual vaccine for this virus, we can do it, but might need a recharge every few years. That's why we have something like the flu virus, where the flu vaccines, where we pretty much have to get a poke every year because they change. They're a different mechanism of how they change, but the variability. Now, retroviruses, I'll just give you a very, very broad hint. They change per replicative cycle. So trying to get a vaccine, it's a 40-year process. It's still ongoing because they change really, really quickly. Please do tell me if this pace is okay and I'm not going too fast. Is this okay for everyone?
Yeah, it's okay. It's okay.
All righty. So I'm going to focus on coronaviruses because that's what we are dealing with in the world today. So what are coronaviruses? They're called zoonotic viruses, like the flu, that's another zoonotic virus. These are viruses that can jump from animal to animal. In fact, there are cases now that the coronavirus has jumped from humans to mink. There are mink farms in the Netherlands. There's one in Oregon. So it just passes from animal to animal. The coronavirus itself is thought to have either a bat or an avian, the ancestor of the coronavirus infected a bat and a bird. We're not sure which came first, one or the other, but they ended up basically creating two lineages. The bat lineage pretty much infected other mammals and then humans, and then it's a back and forth. And then you have the bird, which has infected a few mammals and aquatic species, but we don't have anything that's spread to humans as yet. But this is just the outline of the coronavirus. And how did humans encounter, well, when did we discover coronaviruses, as it were, something that infected us? This is a timeline of how it infected or when it started. So the first human coronavirus was isolated in the Netherlands in 1965. It just caused a bad cold, a very severe cold. And over the next 2 years, there was a flurry of discovery, and through other strains of the coronavirus, human coronavirus was found. All of them just very bad colds. And then things stayed dormant for the next 35 years. And in 2002, we had SARS-1. We now call it SARS-1 because we have a SARS-2, but in the severe acute respiratory syndrome, it affected Southeast Asia. It's thought to have a bat starter, so the bat had the virus. It then moved to a rodent called a civet, and from the civet, it moved to animals. And there were about 8,100 cases. It was limited. The world pretty much deployed, look at the potential, and deployed resources to contain that virus in Southeast Asia. But it still had a high mortality rate. 10% of affected individuals, almost 10% of affected individuals perished from this virus. And then in 2004, 2005, one in Hong Kong, one in England, two other coronavirus strains, not England, I'm sorry, America, two other strains were discovered. And again, bad colds, nothing else. And then again, a period of dormancy when a new virus emerged that was called MERS. That's the Middle Eastern Respiratory Virus. This from bat to camel, camel to humans. The worrisome thing about this was that this has a high mortality rate. About 34% of affected individuals have perished with this disease. This is about equivalent to the Ebola virus and how that kills. And so just a sidebar here. The vaccine, people immediately started thinking of deploying vaccines for SARS-1. The thing is, it disappeared. In 2004, the first SARS infection disappeared before a potential vaccine could be put in place. And because we didn't see any other, pretty bad virus, the vaccine industry kind of stayed dormant with this particular virus until it reemerged with the MERS and the very high mortality rate in MERS. But these are very contained cases. Again, it's not spread globally. Things were put in place to prevent it from going global. And vaccine development is in the works. Now, the problem with MERS is that it comes and goes. So 2012, we had the first wave, it disappeared, reappeared again in 2014, disappeared. And so it comes and goes in bursts. So trying to pin it down is a little bit difficult. And then finally, we have this. We have the SARS-2 virus that is the one that's currently affecting all of us. It emerged in 2019. The first case recorded, I think they were able to find patient 0 was sometime in October of 2019. In a matter of a year and 2 months, it has affected more than 80 million people. Now the mortality rate, as far as we know so far, is lower than say MERS or the SARS-1. However, the infectivity rate is much more. We were lucky in that both SARS-1 and MERS weren't as infective as SARS-2 is. Any questions?
So people are muted, so just the...
No, I think we can go ahead. Yes.
All righty. Okay. So this was one of the first electron microscope pictures of the virus. And it's called a coronavirus because of this. It's kind of, if I were to do it three-dimensionally, if we were to look at it, it would look like the spikes in a crown. And so therefore they called it the coronavirus. And they said it looked like one of the solar flares, as it were. And so they named it as such. Now, this is the SARS virus itself, a diagrammatic representation, and the reason I've put it up there is just to talk to you a little bit about it. This is the genetic information that's in RNA form, and then that is coated in an envelope, and this envelope is mainly protein and lipids. And so I hope you remember way back when this virus started, and still is, wash your hands. Anything you touch, wash your hands. That's because this virus is incredibly susceptible because of this membrane to just regular soap. So just washing hands with soap and water destroys this membrane. If this coat is stripped away, this genetic material cannot survive, will perish. So the whole thing about washing hands, washing everything, that's why we needed it. Now, the other part of it is the spike protein. This is what is being targeted by vaccine scientists, but this is spike protein. Almost all viruses will have a protein on their coat because they need to enter. Viruses, there are plenty of them, but they need to enter our cells in order to replicate, cause infection. So these are kind of like their keys. We'll think of it as a key. And so this is the genome itself. Now, the coronavirus genome is larger than most viruses. It is about 30,000 bases in length, which is pretty large. But it is something that we were able to quickly sequence, and that's why we have vaccines at such a rapid pace. Now, as I said, viruses have very specific hosts. Zoonotic viruses do skip hosts from one animal to another, but most viruses stay within their grouping, as it were. So I'm sure you've heard of swine flus, especially in pig farms in China. They have to cull hundreds, sometimes millions of pigs per year because of an outbreak of swine flu. And they do that because this flu can jump hosts, but for the most part, it is very specific to the pigs alone. And so this is, again, the lock and key mechanism. This is the coronavirus, a representation of the coronavirus, and the spike protein. Now, the spike protein is kind of like a key. The host cell, in order to allow entry, will have different lock points, different other locks, as it were. And within the cell, they have their own keys to have things come in, go out. In the case of the virus, it enters, and because the key is the same, what happens is, this is called a receptor, but we'll just call it a lock for now. The key fits the lock. There we go. This virus can now enter, open the lock, and enter the cell, infect the cell, make lots of copies of itself, and break out. Now, this same bat coronavirus with this key, the human cell has a different lock. This will not match up. So when that happens, then you don't have any infection because the key does not match the lock. It's not going to be able to gain access into the host cell. How did this bat virus come into humans to cause the coronavirus that we now know? Well, the virus changes. This is an RNA genome, and I said RNA genome has no proofreading mechanism. It is going to make copies irrespective of whether it is a true copy or not. If there are mistakes, it's like, okay, let's keep going. We just need to make more copies. And so it will jump. It will keep changing. In the case of this coronavirus, I know the speculation, is it lab made or is it natural? Well, it can be natural, definitely, because these were animals that were packed into a live market, in close contact with each other. And this virus spreads through just droplets. That would be nasal droplets or from the mouth, any kind of droplets. And it also spreads through fecal matter. And when you have organisms all cramped together in a space, it's going to spread like wildfire, and the more options you have to replicate, the more changes can occur. So the current hypothesis is that the bat virus went to a pangolin. The pangolin was infected, then it infected another mammal, probably a rodent of some kind, and then it changed. So the spike protein changed enough that at one point it was able to gain access. So it has a form now that is very similar to the lock. Key can now fit the lock. When that happens, infection occurs. And now the virus can enter a human cell and create havoc. And so this is how the coronavirus ended up from a bat to us, and how we are now spreading it to other organisms. There have been anecdotal references to household pets testing positive for the coronavirus in the US. We definitely know for sure that mink farms, both in the Netherlands and in Oregon, had to be culled because humans have passed the virus on to minks. So with this, a brief kind of introduction. So what happens when the coronavirus enters the cell? And don't worry about all the names. I'm just going to give you a kind of pathway. Here it goes. The coronavirus, the key fits the lock, which means the virus now gains entry into the cell. And once it does, it has the RNA genome. So RNA is just a set of instructions to make proteins. And so protein making machinery in the cell will immediately pick up this RNA. The first thing it does is create a protein that is specific to this virus so that the virus can make more copies of itself. And it generates a lot of copies and instructions to basically assemble the virus again. The virus assembles and then is released. Now, this is kind of like a thief coming in, getting the keys, coming in, completely ransacking the place, using everything. It's like empty shells. Everything is used up in a very short amount of time and then leaves. So what happens? This thief takes everything, including construction rods, windows, doors, support beams. Everything is gone, so the building is going to collapse. In this case, this is just one example, one pathway. Imagine thousands of such viruses attacking a single cell, in which case it's all going to go all the way through and completely destroy the host cell. And as it destroys the host cell, it is going to create more viruses, which will infect more host cells, and the cycle repeats. And so what happens in this situation is that what kind of tissues will this virus affect, coronavirus? This is the troublesome part of the virus. Now, for example, when we talk about the flu is very specific. It only affects the lungs. SARS-1 and MERS only affect the lungs. The fact with the SARS-CoV-2 is that its spike protein, its key, can basically affect tissues in the brain, in the lungs, in the heart, in the kidneys, in the intestine, liver, stomach, and gallbladder, and cause a lot of different infections. So it has multiple entry points, even the vasculature, muscle cells, or things like that. So it has multiple entry points, and that's what is the problem with this. And so people who have recovered from COVID sometimes talk about fuzziness. These are called long haulers. So they talk about fuzziness because it's affected the brain. They have myocarditis, that is heart inflammation, because it's affected the heart. Kidneys can shut down, and some of the symptoms of COVID include a mild stomach upset to severe stomach upset, because this virus can affect the intestines as well. So these are all the different tissues that this virus can affect. It's almost similar to think of the measles. Measles is similar to, it can affect different organs within the body. And so this virus is similar to that. It's not very specific. It basically is broad. Now this is a healthy person has taken in the virus and then infected, and over time damage can be moderate to severe. This is an actual lung that was removed from a 20-year-old in her 20s, a patient in her 20s who after a severe bout of COVID needed a lung transplant. And this is just one of her lungs, and this is what it looks like. I couldn't find a picture of a healthy lung, but imagine a nice rounded structure that you see here that is pink and healthy. This is what it looks like after the SARS-CoV had done its damage. And so that's why it is extremely pernicious, and it's dangerous for people. So I'm going to, before we get to vaccines, this is just a brief history of the virus, look at how we can, a healthy individual, how would we fight this virus? Well, that is, we have an immune system in our, all of us have immune systems that are very effective in killing the virus. So you have cells that infect, and in our systems, we kind of all have in the bloodstream, we have kind of like our security. You know, I don't know, I remember even when I go to visit my sister's house now, at 3 a.m. in the morning, there's this person who walks around, fee, blows the whistle and kind of rattles the door, or fences or walls, etc., to say, I'm here, I'm protecting you. Well, we have a security system. These are professional immune cells. I'm going to call them professional immune cells. They kind of scan our cells, and if they find a pathogen, that means an invader, kind of like the security, they'll start blowing the whistle and saying, there's an intruder here. So that is an activated immune cell. Now this activated immune cell, just like the security, will drag the perpetrators, the invaders, to our lymph nodes, which is like the police station, our major station here. And what will they do? They will activate two types of cells. One cell is kind of just the police force, sending out the brute force police force to say, we have this virus, kill it. These are the T cells. They go out there and they start killing the cells. Meanwhile, these professional cells, these security cells, will also send a message to another group of cells called B cells. Now, this is the armory for our immune system. I don't know, I'm going to refer to James Bond here, but we all know, I hope you know, the reference here is about Q, the person who makes all those gadgets for James Bond. Well, this is the B cell. And the B cell is going to read this virus, deconstruct it, and then go ahead and reconstruct antibodies. And it's going to help the cell remember. These are called memory cells. They're plasma cells. And this is memory cell and added to the database. So our immune systems have an incredible database to say, oh, this pathogen, which means we need this weapon to fight it, which is this antibody to fight it. So what happens is that antibodies are produced and antibodies gum up. If you have the key and you put some chewing gum on the key, it's not going to fit into the lock anymore, correct? That's pretty much what the antibody is doing. It is gumming up and tagging the virus so that it cannot infect the cell. Once that happens, tagging it, we have other cells, immune cells, that will come in and destroy the virus. Now, this is our immune system that works. The problem is, and this is where we come into exposure budget, if we are exposed to, say, about 50 to 100 viruses in a short limited exposure, we're okay. Our immune systems, if we're a healthy individual, should be able to take care of it. Now, that's an easy way to work. That's kind of like if you look at Tamil movies, I remember Indian movies, you have the hero who's fighting the bad guys, the henchmen all wait around and they come one by one, and the hero is able to bish bash and dispose of them. Now, just imagine that in the same movie, instead of the henchmen waiting around, they have millions of henchmen attacking this one hero. And so that's pretty much what happens when we expose ourselves to the virus for more than the given time. So I think right now it's recommended 15 minutes indoors, non-ventilation, can pass this virus effectively to cause a disease, simply because there is just so many virus particles that are coming in. So you have a million bad guys against one good guy, doesn't stand a chance. So we need help for the immune system to get these guys ready to go, to get these plasma, the memory cells ready to form antibodies. And that's why we have vaccines. So we tell the B cells, this is what you have to look for. The B cells will create this imprinted form of memory, add it to the database so that if they ever encounter this virus again, antibodies are produced and the virus is stopped. And so I am now going to, this is innate adaptive immunity, and now we're going to talk about the vaccines that are in play right now for this virus. So we have different types of vaccines. I'd like you to just look at this line here that I'll be going over, and these are the vaccines either that have been approved and are already deployed, that would be the mRNA vaccines, or those that are in development and about to be approved, that includes the AstraZeneca vaccine. And so we'll talk about these briefly one by one. So we'll start out with the ones that have been deployed, and this is the RNA, the mRNA vaccines. Now, in order to understand the mRNA vaccines, we need to know a little bit about the biology of mRNA. So this is a representative cell, one of our cells. Now, within the cell, we have kind of the storehouse of all the genetic information that makes us, and that's the DNA. And the DNA, you can consider this a library of information about us, where the library consists of 46 volumes. That would be the 46 chromosomes that make us up. Here's the thing. This DNA, the volumes, those books will never leave the nucleus. They always stay inside this protective bubble. So if instructions have to be made, say to make an antibody or a specific protein, right now I'm talking to you, that's a series of proteins in my system that are being made. You're listening to me, a series of proteins. So we're making proteins all the time, the instructions of which are in our DNA. but the book never leaves the library. So what we can do is make a copy of a set of instructions, and that copy can then be sent into the cell so that the workers can make the protein. Now, this is many types of RNA are made in the nucleus. One such is the copy of instructions from DNA, and it's called the mRNA. Now, this environment, the cytoplasm, the outside of the cell, is extremely toxic to mRNA. Pretty much it gets eaten up as soon as it enters in. So it needs to be protected, kind of like laminating paper. So in this case, we have to add a cap, a kulla, and a tail. In Tamil, we call it val. So we need a kulla and a val, basically, to protect this in the cytoplasm. And then, so we go ahead and the nucleus adds this on, and only then do the gates, we have gates in the nucleus, only then do the sentries at the gates allow this virus to, allow this mRNA to leave the nucleus and go into the cytoplasm. Once it's in this cytoplasm, we have, and this is a one-way ticket, by the way, lots of RNA are made in the nucleus. Nucleus can only go one way, from the nucleus to the cytoplasm. It cannot go from the cytoplasm back to the nucleus because the gatekeepers here will not allow it back in. DNA can re-enter, proteins can enter. However, RNA, once it leaves this safety cocoon of the nucleus, cannot return. Now, once it's in the cytoplasm, it's very short-lived, and as soon as possible, protein-making machinery will come in, start reading the instructions, make the protein required, including antibodies if required, and it does the function within the cell or is exported out of the cell. But this is the basic function of mRNA. So based on this, we were pretty lucky in that we have the tools now to go ahead and sequence the entire genome of SARS-CoV-2. And this was done in, believe it or not, January, I think the sequence came out on January 29th of this year, 2020. And by March 16th, Moderna already had its first candidate vaccine from the mRNA. Now, the technology has been in play, has been theorized, hypothesized from the 1990s. By 2010, people were beginning to say, let's do this, let's work with other, because it's a very nimble way of making, a nimble way of making a vaccine. And even if the virus were to change, we can go ahead and effectively change with the virus just as quickly because it's very simple with the tools we have. So what are the tools? Scientists basically looked at those keys, the spike proteins, the antigens as they're called, of this. They found what the sequence was. And this particular virus, COVID, SARS-CoV, the one saving grace for vaccine scientists is that this mRNA doesn't have to be, it's called positive strand because the cell can directly read it. It doesn't have to change conformation. It's not backwards. The flu virus, for example, is a minus RNA that is backwards. So this basically, if we're doing any immune vaccine for that, we have to correct the words, we have to turn it, reverse everything. Here it's just straight up. So they focused on the spike protein, the coding region for the spike protein, the spike protein information. They literally just cut it out. That's all they had to do. Now, this is mRNA, and I just said that the mRNA for the cell is extremely hostile environment. They have to protect it. They have to laminate it. So this is what scientists in Moderna and Pfizer did. They added that cap, they added the tail, and now they had to think of a way. The first trials, they tried to inject it directly into patients. It wasn't taking effect because there's no lock and key. We have very good security systems in our cells, and so this particular mRNA couldn't enter the cell. So they had to package it in a delivery vehicle. They needed to put it in needed a delivery mechanism. And so this is a lipid nanoparticle. It doesn't have any extra proteins. The lipid layer looks very much like our own cell membrane. And then they went ahead and introduced the mRNA, multiple copies of the mRNA of this particular spike protein with the attachments included, and then went ahead and injected it. So this is a graphic of how Pfizer pretty much did their work, which is basically you have the spike protein, they took that RNA information, cut it out, placed it inside a delivery mechanism, that's the lipid nanoparticle, injected it into the cell, which triggered an immune response, which I'll get to in the next slide, but triggered an immune response. so that antibodies were produced and also T cells were deployed, because the cell now made the spike protein itself. It's teaching the cell. Usually vaccines were kind of showing the cells in our system, our immune systems, this is what the bad guy looks like. Now we're actually teaching the cells to make their own version of the bad guy, the pathogen, so that the immune system can learn from them. And so the antibodies are produced as well as T cells deployed to kill these infected cells. We don't want the spike protein, as it were. That's the reaction that we have. It's pretty much typical of all any vaccine we get. I'm sure, I know my mom used to say, if you're giving a child a vaccine, give them some, first of all, give them an acetaminophen, I think we call that paracetamol in India, just to reduce the fever, because it is coming. There's going to be an immune reaction. And so this is what happens. Now, I'm also now going to switch to Moderna, which used the same mRNA mechanism. And so what happens pretty much is that, again, they have this information that was the mRNA for making the spike protein. They fitted a cap and a tail, put it inside a lipid delivery mechanism, and injected it into patients. Now, it has two ways it can go. It can either infect a regular muscle cell, where the mRNA comes in and then the protein making machinery immediately starts reading, makes the protein, and then the protein will then go ahead and form the spikes, as it were, in the native cells, in our own cells, and triggers an immune response. It can also, because it's in the muscle, we have our lymph nodes right about in our underarm region here. Those lymph nodes, this particle can go into the lymph nodes as well. These are our professional protectors. These are our professional immune cells. They will go ahead, read the protein, break the protein apart. This is our armory, as it were, and then see what kind of antibodies have to be produced in order to block the virus. So two mechanisms take place. Now, this approach with the mRNA vaccine, very simply put, is that it is incredibly quick. We just need to focus on that spike region or the antigen region on the surface of the virus and just crank out the vaccine itself. Like I said, it took less than 6 weeks for Moderna to go from the sequence itself to actually creating the vaccine, the first version of the vaccine, and sending it out to be tested for clinical trials. Now, this had never been done before. Again, on paper, it sounded great. So they had to do testing, trials and testing. But it seems to be with the efficacy, there's 95% efficacy with both Moderna and with the Pfizer vaccines. And so that seems to be highly promising. Now, a caveat here is that no vaccine is going to be 100% protective. All of them, including the smallpox vaccine, only had about 80% effectiveness. But we'll get to that in a bit. Herd immunity is developed and the virus has nowhere else to go. It will ultimately die. In this case, the mRNA vaccine, the pros, and I'll get to that, is that it's incredibly fast to make. The big thing is there are no infectious particles. It is never going to cause an infection. It may cause an immune response, slight elevation in temperature, and we're seeing some cases where people who have allergies do have a more severe response. But for the most part, this is not going, there's no infective particle in here. So this seems to be the fastest, most effective way that we know of right now. Now we're moving into Oxford AstraZeneca. And that's the next one up. These are the viral vectors. This is another new platform that has come into play. And so what does this do? Viral vaccines, vector vaccines, are basically non-pathogenic. You alter the virus. The virus's infective materials are removed. And then we take, this time we have to convert that RNA sequence to a DNA sequence. And that DNA sequence is incorporated into the delivery, into the package, as it were, the delivery van. And then that is then administered as a vaccine to the recipient. And then the recipient mounts an immune response. Again, the cells, all of these mechanisms are teaching the cells to make that spike protein so that the immune cells can go ahead and recognize what the intruder looks like and can then mount an immune response to help. And so this is the actual Oxford. So what did they take? They needed a delivery mechanism. So they took the chimpanzee adenovirus. That is a virus that infects chimpanzees, but has a very similar key mechanism to some of the locks in our cells. and then removed the pathogenic part here. So now it's just got the keys, but no actual viral component. The other end, they took the SARS-CoV-2, they studied again the spike protein genome, they basically converted it to DNA sequencing, and we can do that pretty quickly right now, and then inserted the spike protein information into the adenovirus that had been, that is no longer infective, and then injected into the host. Now this will go into the host. DNA can enter the nucleus, so it is going to enter the cell, and then it is going to give instructions to make the proteins, the spike proteins, and then an immune response is mounted. The body produces antibodies, and so when the individual is exposed to the actual virus, the immune system already knows what to look for and will then go ahead and immediately shut it down so that it doesn't infect anyone else. So these are the adenovirus, the viral vector vaccines. Now the downside to the viral vector is that these spike proteins, our immune system, we have used adenoviruses for a few others, so repeated use. Right now, this is a novel use, this is in works, but these, we only have a certain set of adenoviruses, meaning a certain set of keys that can enter the locks of our cells. So our immune system is already going to be trained. Yes, it's trained to do the spike protein, but this is also a foreign protein, and the immune system will recognize this. And so the next time we use a viral vector, the immune system may shut it down even before it can actually deliver its package to the cell, which is the genetic information to make the spike proteins. So that's one caveat behind that. Basically, our system's going to say, oh, bad intruder here. The package is not the best, and so it's going to go ahead and shut it down. So that is something that we are, that scientists are considering when talking about these viral vectors. However, the big thing about this is that it's a lot more temperature stable than the mRNA. mRNA is inherently a very unstable molecule. It is going to degrade quickly, and that's why it needs those very low temperatures of -90, -70, etc. Whereas in the case of the adenovirus, the vector viral vaccines, just a regular refrigerator, 2 to 8 degrees centigrade, which is our refrigerator temperature, is more than enough to keep it stable. So for countries like India, this seems to be a more safer way and more effective way to vaccinate. So this is with the viral vector vaccines. Now others in development include Johnson & Johnson, that's also using an adenovirus. It has a lead candidate, they're in trials. And in China, the CanSino Biologics, they're also using an adenovirus with the S protein DNA put in there to go ahead and teach our cells to make, get an immune response, and to teach them how to get the cells. And then we have another idea. Remember I told you just a few minutes ago that an adenovirus, over time, we may develop immunity to the van itself, to the package deliverer itself, and say, oh, bad delivery mechanism here, this is a bad guy, destroy it. So Sputnik, that's, I think Reddy Labs in India are doing that. They have a 2-vector vaccine, and that's essentially, the first vector is, this is one van, think of it this way, that's delivering the package. So the first package is delivered, the immune system creates antibodies. Now it has to remember, so it's, sometimes we all need, even for exams, we're reading up again. Why? Because we need to remember, we need to jog our memory. So just to do that, Sputnik uses a second viral vaccine, and this one will go ahead, a second package, a second delivery mechanism, as it were, delivery van, think of it that way, and that will enter the cell, creates the same antibodies. It's kind of reinforcing the learning, as it were. And so this kind of eliminates the possibility that our immune systems will recognize the first delivery van and destroy it before it can deliver the package to the cell. So they trick it by sending a second delivery van with the same package, but this time the immune system cannot recognize it, so the package is delivered. And so this is the Sputnik system. Now, initial trials have shown that the Oxford AstraZeneca virus is anywhere from 70 to 90% effective. Sputnik has been registered to be, I think, 92 to 96% effective. However, data comes from a very restricted source. Other scientists are not able to study the data. And so with the Sputnik vaccine, it's still kind of on hold, as it were, trying to figure out how effective it might be on a global scale. So these are the viral vector vaccines. Another one in the pipeline are the inactive viruses. And these are mainly in China. So what does this do? You have a virus. So an adjuvant is kind of a chemical, something like aluminum, or I think aluminium. I'm sorry, I've become too American here. It's aluminium is used to effectively transfer the genetic information required. So what it does is this virus is destroyed inside. All it has is the spike protein for the coronavirus. So the RNA genome is completely destroyed. So this spike protein goes into the cell, and then it will go ahead, and these are our professional immune cells. Remember the security guards? Security guards will pick it up, read it, activate it, and then take it to the necessary immune cells to either destroy it or to make weapons to destroy it at a later date. And so this is the inactivated viral vaccines. They're still in production. Clinical trials have not yet, they've started, but they're still only in phase 1. They're still working through the other phases as of right now. So Sinovac, one of the drawbacks of this, in fact, one of the drawbacks of the viral vector vaccines and the inactive vaccines is that production time is a lot longer than the mRNA vaccines. So this means a lot of virus has to be generated, and a lot of virus that is generated will then have to be purified so that it can go ahead and be used as an effective vaccine. So that's one of the downsides to that. Now, another type of vaccine that's in play is a DNA vaccine. There's a lot of them. So in this case, we again take the genetic information of the spike protein and we introduce it into the DNA form, into something, a small circular piece of DNA called a DNA plasmid. Now, the problem with this is introducing it into a cell. When they tried directly introducing it, didn't work, because there's no delivery mechanism. So scientists, vaccine biologists, this is what we do for bacteria if we want to introduce a DNA plasmid, is electrocute it. So a tiny electrical pulse is sent to our cells that creates a small opening, and that opening is enough for the circular DNA to go in. Now this circular DNA goes in, oops, sorry, I have to go back here. This circular DNA goes in and it goes directly into the nucleus. There it's converted from DNA to RNA. The RNA then leaves the cell and then it goes into make the spike proteins that can then either, again, trigger the immune response and then create a pathway. So there are two pathways. This is in a regular cell. This is in our immune cells. The immune cell literally takes it, the protein, and breaks it down and creates the necessary antibodies, teaching our immune system what to look for. The one possible downside, one thing is that we're not dealing with any infectious particle. It's just the spike protein information. The downside is, and they're still studying, the possibility of this DNA, because it's going into our nucleus, integrating into our genomes. Now, there's not enough studies to say that that is happening, but there's not enough studies to say that that is not happening. But this is another delivery mechanism for a vaccine. And then we have the last, I promised, the last type, and that's called subunit vaccine or protein vaccine. In this case, they literally took the spike protein, they packaged the spike protein in a liquid nanoparticle and injected it into cells. And then the cells, this breaks down, the cells see this protein particle, the immune system comes in, recognizes it as an intruder, takes it into the armory cells, make antibodies to go ahead and destroy it. And so that is another mechanism that's being worked at. Now, the problem with this is we need to scale up. We need a lot of these spike proteins in order to generate an effective immune response. So again, this is going to take some time. Okay, so someone had a question here. Mr. Bharat had a question. Viral vector method, how will a second dose work if the body recognizes? That's why they're thinking about the 2 virus system. And it takes a little bit of time for our immune systems to recognize. That's the worry with the viral vector method. And so Sputnik has the 2 viral methods. So yes, Oxford does say that they need 2 shots, and that's to reinforce. And they're hoping that the second, so right now when Oxford does it, that's why they haven't released the data yet, because initial data was very confusing for the Oxford virus. I'm not sure if you read about this, but the first time when they published everything, it looked as if a single dose was way more effective than the 2 doses. They had almost 90% efficacy with a single dose, whereas only 60 to 70% with a double dose. And then they discovered that they didn't keep good records. And so that's why they've had to go back and redo everything. What they found is they mixed up. They didn't give the second dose to younger people. They only gave it to older people. And so is that because they're giving it to the the higher age group, the 65 and above, they gave a second dose? Is that the virus, the dosage itself was not effective? And then they didn't give it to the younger population, and was it because the younger, healthier population had some kind of immunity or immune response to it already? So they've had to go back and they've had to redo some of the trials just to get their numbers. Now the science behind it is still valid, but it is the numbers were a bit skewed. And so what is the guarantee that the sequence of things after vaccination? Oh, that is the other thing that I will be talking about actually, because you do need to, even after vaccination, it's not going to be immediate. We can't get a vaccine and immediately go out without our masks and forget social distancing. It's a process, and it will take a little bit of time. But this is the protein subunit vaccines, and this is another deployment method. And then, so this is just a quick table to tell you about the different vaccines that are in play, how effective they are. And as I said, this number here, they're still working on it because of their mix-up with the records and how their trials were run. And then Moderna and mRNA, the mRNA vaccines have 95% efficiency, and the Pfizer has to be at -70. Moderna, you can keep it at -20, which is our regular freezer temperature for up to 6 months, and it stays stable. And then we have the Sputnik, and this is in dry form. In powder form, it can stay at regular temperature, fridge temperature, and so that is a little bit more effective. Now, this we have enough evidence to say that it is a little bit, yeah, it is more effective. So Oxford, I think the Serum Institute of India is actually following the Oxford Oxford University AstraZeneca vaccine, and Bharat, I think it's called Bharat
Bharat
Biotechnology.
Biotech, yes.
Yeah, Bharat Biotech. They're using a live attenuated virus. They're doing the virus that's very similar to what we get for polio. It's inactivated, a weakened virus to teach the immune system. But that's still under study. But right now in India, the Serum Institute, the Oxford AstraZeneca, they're ready to deploy, and the Ready Labs are making the Sputnik V, that's the Russian viral vector vaccine. Now, we do need to have the second vaccine. So if you miss the second vaccine, if they say after 3 weeks you need to get the second vaccine, just get it done as soon as you remember, kind of, as possible, because that second vaccine is required. So if the first one, if somehow somebody, forgets after the first one, oh, it's 3 weeks later, but I missed it, don't worry if it's a week later or 2 weeks later, I think even a month later, just get the second vaccine. It's kind of like a booster to help our immune systems, just remember a little bit better what to look for. Now from here, I am going to just give a brief table about the advantages and disadvantages of the different vaccines. Now, the DNA and the RNA and the protein, all of them have one thing in common: no infectious material, and they can be used on individuals, especially the DNA can be used in immunocompromised people. We're looking now at the RNA vaccine, and we're finding that people who have significant allergies, including shellfish allergy, pollen allergy, people who have allergies do have a more severe response to the vaccine, although nothing has been fatal so far. It's immediately treated. That's why now people who are given both Moderna and Pfizer have to take the vaccine and then wait in the clinic for 15 minutes to make sure that there is no adverse reaction. The good thing about the RNA, as I said, is that it's a rapid design production and it produces antibodies effectively. Both DNA, RNA are very easily scalable. In the case of DNA, variable immunity, it does require the host delivery special delivery mechanism, and there's the potential risk for host cell integration of just the plasmids of this spike protein. In RNA, it's basically the storage and the delivery mechanism. Now, scientists have been able to find a sensitivity of all these vaccines. So comparison of comorbidity sensitivity, basically individuals who are not immunocompromised or do not have comorbidities, the DNA vaccine, all these vaccines work very well. The RNA vaccine seems to have effects on people who have allergies, but it seems to be fine with people who have comorbidities, simply because it does not affect that pathway. It's a whole different pathway, which I'd be happy to go into. That's the gate system, why we have these comorbidities and why this virus is attacking people with comorbidities like high blood pressure or diabetes. In the case of proteins, again, no infectious material, strong antibody response. Identifying be more efficient, or is the current scenario, in fact, is for this RNA, the current scenario that we have has been highly efficient and much, much faster. To put it in perspective, it took us 40 years to develop an effective polio vaccine. It's taken the scientific community about 9 months to go from here's the virus all the way to here is the vaccine for it. And so these are the different types. The viral vector method, the adenovirus, right now cannot be used for immunocompromised people. The other problem is, although it's supposed to be an inactivated virus, we're not sure how good that inactivation is. So there's a risk of the virus regaining virulence. That would be the vector, the packaging material. And scale up, for proteins, the viral vector and the inactive virus, scale up, going from a small batch to the millions of doses required can be a little difficult to work with. Okay. And so I'm going to talk about, I think someone asked, what happens if we get the shot? Well, if we don't get the shot, just going to go ahead and minimize that. If we don't get the shot, well, onset of infection, you have healthy individuals and this spreads. Now, this is not nearly as infective as, say, the measles. This infects, if one person gets it, another 2 people can get it, kind of. And so, but the infection spreads very quickly. And then you end up with people who either have immunity because they recovered, but you have a lot more people that can also die because of the disease, the severity of the disease. Now, without immunization and social distancing, we are creating something like a circuit breaker. It's kind of, there's a break. There's no connection. So if there's a break, the virus cannot go from individual A to individual B. But even then, you still have higher mortality rates, and you have just a few people who have immunity. So these potential dark blue people can be infected as well. And then if you have immunization and social distancing, the vaccine is acquired, people are still going to be infected. So having the vaccine means potentially a person who has the vaccine is not going to have symptoms because it's going to shut down, but they can still carry that viral load until the immune system is able to completely suppress it, which means that if they are in close contact with someone else, they can pass that virus on. Basically, with immunization, we are creating an artificial circuit breaker. This infective individual is now this healthy individual is protected by these 2 immunized individuals. So these 2 individuals are not going to affect this person or these people here, kind of creating a buffer to protect people who have the infection. This person is probably going to infect this person because this individual was not immunized. Over time, it will go ahead and spread through the population so that the virus has nowhere else to go. And that's what we're trying to do here. So for example, Ebola is a virus that comes and goes. Why? Previously, it would be in this small village. and only in the village. So it would run through the village, it would infect everybody in the village, but because nobody was moving out of the village, that virus had nowhere else to go. People either died of the disease or had immunity to the disease. The virus would disappear. It would reappear again when the next contact happened, but that's how it worked. Now we live in a global world where we can get on an airplane and spread the virus pretty much everywhere. So we need the vaccine in order to prevent that, so that we create this buffer, as it were. And even with the vaccine, until a significant population of the world is immunized, we still have to maintain social distancing and wear our masks. So the safety of going out is better, yes, but still maintaining the mask wearing and the social distancing is required. Now, when I talk about this, as I'm talking about it, it is virus, there are more variants coming in. 2 variants that have come up that have seemed to be highly infective. They've taken over the majority of new cases, are the variant in the UK and the variant in South Africa. Now, the UK variant, 23 mutations were observed in the virus itself, the whole genome, including 1 in the spike protein coding region. Similarly, the same mutation that was found in this spike coding region was also found in the South African variant, but the South African variant had a few more mutations. The one thing is, when they designed this, the spike protein, the information, the key is still the same. The key still needs that same lock. So the key is still effective. If we can block that key, then we can still have immunity. So these vaccines are still effective. How long they will be effective, however, we don't know. So long term, just having the vaccine right now, it might be something that, one thing that we first observed was this virus was not mutating as quickly as other RNA viruses do, but now we find that it is accumulating. And the reason is that every time this RNA molecule replicates, it's going to make mistakes. There are going to be mutations. Now, if you give it enough people to infect, it is going to create more and more vaccines. This is just the defective replicative mechanism. Now, our DNA, when it replicates, makes almost perfect exact copies because of the proofreading mechanism. However, RNA does not have that proofreading mechanism. So it is just going to make out the sequence and put it together without checking to make sure that it is an exact copy. So changes are made. And there are certain regions where it really wants to crank out quickly. It doesn't check. It doesn't go back to check. So those changes happen. And so you have more people infecting, more replicative cycles, more chances for mutation. And I'm sorry, that was quite fast, but I would be happy to answer any questions I do have if you want to know specific questions. We don't know how SARS-1 died out. That is the mystery that we would love to know. It just disappeared. And we don't know whether the containment measures, pretty much Southeast Asia, Taiwan, Hong Kong, Vietnam, Laos, those regions were just, and South China, southeastern China, completely locked down. Wuhan, they didn't do that. When the first virus came out, they decided to not go ahead and shut everything down. And because of the global world we're in, people started moving around. And just to put this in perspective, 2 direct strains, the most effective vaccine, the RNA vaccine is good. The viral vaccines the vector vaccines are also good. We're still gathering data. Then we know what to do with. So we're still gathering data about this to figure out what it is that we can, which would be most effective for individuals, and going from there, as it were.
Mallika,
A couple of more questions.
To stop sharing screen.
Oh, yes, I can go ahead and stop sharing. There we go.
Yeah, a couple of more questions.
Sure, go ahead.
Yeah. Now, here in the chat box, what type of flu was the Spanish flu of 1918?
That was the influenza virus, actually. And it's called the Spanish flu, but it shouldn't be called the Spanish flu. It actually, we've sequenced it and we found that the origin came from China. It's called the Spanish flu because 1918, World War I, when it first started appearing, it started appearing in 1917. But World War I, and it started appearing in Europe, but World War I was ravaging through the world. So the news headlines all over the world was what's happening in the war. Spain was neutral. And so Spain did not worry about the war. Instead, they were worrying about this virus that was infecting them. And so what did they do? They called it, their newspaper said, oh, this person has the virus, our king has the virus. And so they made it the big virus thing, and it became the Spanish flu because Spain was the first one to ring alarm bells about the virus. But it is an influenza virus. The influenza virus that affects us every year now is actually a great grand descendant of that 1918 flu virus.
Thank you.
So.
Thank you.
There was also a little bit additional thing, right? If I can add, perhaps you can confirm if I'm right. In addition to people focusing on the war, Germany and France did not want the news of the infestation to go out because that would spoil the morale of the troops. And in fact, the troops are the ones who actually had this quote unquote Spanish flu worse because they were in trenches. Those days they fought wars in trenches. So they're all packed together. So it's also, they put a lid on the information from what I remember reading. So nobody knew that
they just
those guys
to
had
spread
it.
out there. Pretty much what some of countries did here too. I mean, we still don't know the accurate numbers from some places in the world right now,
Right.
to say who's affected, who's not. But yeah, they wanted to suppress. The war was the big thing. And that's how it came to the United States, because all these returning soldiers were packed into ships, thousands upon thousands, and the virus had lots of people to infect. It came to the US and it had even more people to infect. And it was global because troops were returning home. It was spreading everywhere.
Okay. Some of the things that have come out of the chat box are not questions.
And yeah, we don't know the efficacy of the vaccines right now. The Pfizer vaccine, I've had friends who've taken the Pfizer vaccine, and they are basically reporting back to me that I had 2 people who confirmed for them, at least it was milder than even taking a flu shot. One person to say that they had soreness in their arm, but otherwise they were okay. So individuals without a history of allergies, actually, this vaccine seems to be not that reactive, as it were, but it is creating antibodies, and they'll have to take the next shot.
How will they organize a delivery mechanism in India for a vaccine which is to be stored under very low temperatures?
Exactly. So for that, India, I think, that's why the Serum Institute is focusing on the viral vector vaccines, because that can be stored at refrigerated temperatures. And even in the US, actually, rural areas don't have -80 freezers. And so delivering this vaccine for them is, it's basically a logistical, I mean, they're shipping these freezers to those regions right now. The Army is doing that in the US. The armed forces are doing that because even rural areas don't have the -70 that's required. The good thing about these mRNA vaccines is it's really quick. We want to generate a new vaccine, we can go ahead and do that very quickly. The bad news is that it is so highly unstable that we need to keep it at really cold temperatures. Moderna did wait a little bit. That's why their vaccines came a little bit later. They reformulated so that it can at least be stored at -20. Long-term, -80 is good, but at least for short-term, it can be stored in a regular freezer. Can the mutation, meaning the viral mutation, if it is the virus, it's not, because it's an mRNA genome, and mRNA is, as I said, it's always going to be one way. It can go from the nucleus outside, it can never re-enter the nucleus, and any information that is stored will have to be in the nucleus. That's the library of the things that make us us. So a mutation can happen. The mutation does happen inside our bodies. When that virus replicates inside our cells, it is making mistakes. Those mistakes are packaged, goes on to infect another cell, and so more and more mistakes can accumulate, but it'll never be part of our cells, as it were. It's just going to destroy our cells.
Thank you. Thank you very much. Any more questions?
We don't know the long-term effects.
Long-time effective vaccines, yeah.
The vaccines, we don't know yet. That is something that so far that they've studied, it doesn't seem to have the the mRNA vaccines especially, there's no viral component to it. It's just genetic information. That is, again, mRNA does not last very long in our cells, so it's not going to have any vestiges as far as we know. Long term, it's kind of like the GMO foods. We need GMO, genetically modified foods, because we have an ever-growing population on this planet that needs to be fed. And after years, we have discovered that it is okay. We're not going to, so far, no adverse reactions, but it's something that only time will tell.
Yeah, yet again a question. Safety protocols as robust in quick vaccine. Bring up you can see the question, yes. Yeah.
Bring up cycles as compared to traditional cycles. This was a little different because vaccines to get approval takes years. That's why 2010, the first mRNA vaccine was proposed. People were going to put it into work actually in 2019 when everything shifted and it was all COVID all the time. And so it was, they've done trials, they've done as many trials as they can. It's about as effective as it can get. They've done a multitude of trials there. But again, maybe this is how vaccines will be produced from now on. The world will have to react to the next pandemic and be nimble on its feet and create the next vaccine. But it seems to be effective right now. Now, the quality of the data available on the vaccines, both Moderna and mRNA, the Pfizer vaccines, they have a lot of literature. They're being very transparent in what they're producing. AstraZeneca, the Oxford AstraZeneca was the mix-up with the numbers. That's why there's a delay in approval for that vaccine. They need to get their numbers right. But again, they're being transparent about it. So far, the Sputnik vaccine, as far as I know, I was actually looking for some papers on that. We just have data reported by the Russian government. We don't see the actual numbers. They've given numbers, yes, but they haven't had it peer reviewed, as it were, so that's still there. The other vaccines, we're still collecting data. They're still in initial phase trials, so we're not sure what the quality of the data is for that.
Right.
Okay.
I think it's a super presentation. One of our members, I think, Balu, he has spoken for all of us. He says even oldies like me can easily understand. That goes for all of us. Most of us. All of us in Artistry anyway. Yeah. Bharat?
Yeah, I had one quick question, but Mr. Subramaniam was going to follow.
No, go ahead.
No, the quick question was, we've heard of CRISPR. Did it have a role? Is that the sort of the standard way to do these these days?
CRISPR was not used for this. We have specific chemicals, enzymes that can cut and paste. So CRISPR was not used for this mechanism here per se, but they are molecular scissors. They're slightly different. They work for slightly different things. But from what I understand, this was just using straight up enzymes to cut out specific regions and then paste them. But we can use CRISPR mechanism for future reference. If we can't find the exact scissors needed or the exact enzymes needed, we can use CRISPR because they have the guide RNAs. We can say, this is what we're looking for, find this, and then cut it out. But for the RNA molecule, right now, they're just using the cut and paste with the enzymes.
Hello, can you hear me?
Yes, I can hear you.
This question is related. Sorry, I'm just going back a little. If I'm too loud, please tell me.
No,
go ahead.
When I asked something about the mutation, you said the sequence could go wrong and produce the mutation.
Yeah.
What is the probability the same thing cannot happen after the vaccine is introduced inside?
No, that is basically the vaccine. It is going to prevent the virus from entering the cell. So the antibodies will prevent, it's going to kind of block off all the spike proteins. So it's not going to be able to lock onto the cell. And so it's not going to infect, it's not going to be able to replicate, which means that if we've effectively blocked its replication channel, it cannot make mistakes when replicating, and therefore you cannot have mutations beyond that point.
No,
it's only when the virus enters the cell and it begins replicating that it starts making these mistakes.
It's a last question from me anyway. I've been asking a few already. The mRNA, it touches the genetic code, correct? Has anyone studied the effect of changing any genetic code and its lasting effect in the literature? I could find many.
I'm sorry?
I tried to find something about, but I couldn't find many references towards this particular domain.
Okay. Now, genetic code, if we want to talk about ourselves, that's the DNA, the information that makes us us. I don't know whether you heard about this Chinese scientist who used CRISPR mechanism to confer HIV immunity to two embryos, twin girls. And what they found was that this person is now being debarred from science, completely barred from science, cannot do any science. Essentially, the long-term effects is that by changing one aspect of the genome, they changed other aspects. Now, this is in DNA. RNA is just a very temporary, it's just like a piece of paper that is easily chewed up in the environment. So I don't know, just to give a reference here, I remember my mother had to laminate my birth certificate. I'm not that old, but the paper that the birth certificate was on began to get chewed away. And so imagine that in a really rapid time frame, that it is just chewed away. As soon as it enters, as soon as the mRNA enters the cytoplasm, the rest of the cell, there are enzymes in the cell that are busy chewing it away. And so it doesn't last very long. And again, mRNA, any RNA is just one way. It goes from the nucleus to the cytoplasm. It cannot go back into the nucleus. And so changes in the mRNA can only happen when DNA codes for it. So there's a fundamental change in the DNA that's going to change the RNA output, and that will then carry forth with whatever protein is produced. However, the mRNA on its own, when it's a viral thing, it is going to change over time, but that happens, nothing to do with us, nothing to do with our nucleus and the information that makes us us. If
Can I have one more
answers your
question?
Sorry, go ahead.
Can I have one more question?
Yeah, can we? Last question, yes.
Last question. See, why statements regarding the commodity effect are so fussy?
Commodity effect.
Nothing is very clear about comorbidity, effect of comorbidities.
Oh, comorbidity.
They are very fussy and often misleading also.
Yeah, that I have a slide for. I don't want to confuse anyone, but basically the key to the lock for our cells, the coronavirus, human coronavirus, goes to specific receptors. These are called ACE2 receptors, and they are important for regulating blood pressure and also kidney functioning. And so they're everywhere in our systems, and therefore people who have a tendency for higher blood pressure have a few more of these receptors, and therefore there are a few more locks for the key to go in and enter the cell. Therefore, they have a higher incidence of infection. Again, people who have diabetes, their liver cells, their pancreatic cells are also not, they also have more of these ACE2 receptors, again, blood pressure issue. And so there are more locks for the keys to go in and unlock and enter into the cell.
Thanks a lot. Nice presentation.
Thank you. I must resist the temptation of prolonging this session because we have to release Mallika. It's about 7 o'clock in the morning there?
It's 7:15 in the morning.
7:15, and you got to get ready and go for work. Right. May I request TSS, TS Subramaniam, or Atashian to formally thank Mallika? Please come in.
Yeah. Can you hear me?
Yeah.
Hello, you can hear me?
Yes, of course.
Yeah. Last 1 year has been really very trying, and there was a subspeed of information all over the social platforms, confusing a lot of people. Now, of late, the one more added confusion about vaccines, how they're going to be useful, and it has created a lot of fears in the mind of people. Dr. Mallika's lecture today has put at rest all these fears and confusions. She has given an excellent overview, starting with basic DNA, RNA retroviruses, examples of smallpox, coronavirus, and HIV. And she went on to explain adaptive immunity T cells and viral vector vaccines. She covered a whole wide range of topics within a short span of time. And it's been a very excellent talk, very lucid and simple for a layman to come to grips with the subject. Thank you very much, Dr. Mallika, for an excellent lecture on
Thank you for
COVID-19.
inviting me.
Thank you.
Pleasure. And
Thank
I'd
you
be
very
more
much,
than happy
Mallika.
if you have any questions. I'd be more than happy to answer them. My mother can share my email, and I'd be happy to answer questions you may
Wonderful.
have.
Wonderful. We shall forward them to you, or to your mother, and then
Yeah.
hope to hear from you. Thank you very
Thanks
much.
very much. Thank you.
Thank you, every one of you who's come and been here with us. On that note, let me close this meeting.
Okay.
Thank you. Thank you very
Thank
much.
you.
Mm. Mm.